Definition of toxicology. Here we'll discuss the definition of toxicology. Toxicology can be defined quite simply as branch of science that deals with poison. Broadly speaking, a poison is any substance causing harmful effect in an organism to which it is administered, either deliberately or by accident. Clearly, this effect is closely related since any substance at a low enough dose is without effect, while many, if not most, substances have deleterious effect at some higher dose. Much of toxicology deals with compounds exogenous to the normal metabolism of the organism. Such compounds are referred to as foreign compounds or more recently as xenobiotics. However, many compounds endogenous to the organism, for example metabolic intermediates such as glutamate and hormones such as thyroxine, are toxic when administered in unnaturally high dose. Similarly, trace elements such as selenium, which are essential in a diet in low concentrations, are frequently toxic at high level. Wherever the harmful effect of physical phenomena such as irradiations, sound, temperature, and humidity are included in toxicology, appear to be largely dependent on preference of writer. It is convenient, however, to include them under the broad definition of toxicology. The method of assessing toxic effects is another parameter of considerable complexity. Acute toxicity usually measures as mortality and expressed as LD50, the dose required to kill 50% of a population of organisms in questions under specified conditions is probably the simplest measure of toxicity. Even so, reproducibility of LD50 value is highly dependent upon the extent to which many variables are controlled. This includes the age, sex, and physiological condition of the animal, their diet, the environmental temperature and humidity, and the method of administering the toxicant. Chronic toxicity may be manifested in variety of ways: carcinomas, cataract, peptic ulcers, and reproductive effects, to name only a few. Furthermore, compounds may have different effects at different doses. Vinyl chloride, a potent hepatotoxic at high doses, is a carcinogen with a very long latent period at low dose. Most drugs have therapeutic effect at low doses but are toxic at higher levels. The relatively non-toxic acetyl salicylic acid with aspirin is a useful analgesic at low doses, but it is toxic at high doses and may cause peptic ulcers with chronic use. Considerable variations also exist in the toxic effect of some compound administered to different animals, or even to the same animal by different routes. The insecticide malathion has a low toxicity to mammals, whereas it is toxic enough to insects to be a widely used commercial insecticide. The route of entry of toxicant into the animal body is frequently oral, in the food and drinking water, in the case of many chronic environmental contaminants such as lead or insecticide residue, or directly as in the case of accidental or deliberately acute poisoning. Other routes for non-experimental poisoning includes dermal absorptions and pulmonary absorption. The above routes of administrations are all used experimentally, and in addition, several types of injections are also common: intravenous, intraperitoneal, intramuscular, and subcutaneous. The toxicity of many compounds varies tenfold or greater depending upon the method of administration.
Early developments in toxicology. Soon, nature and magnitude of toxic effects were studied. Factors like physicochemical properties of the substance, its bioconversion, the conditions of exposure, and the presence of bioprospective mechanisms subsequently dominated the scene. Morphological and biochemical injury produced by a toxin was classified as inflammation, necrosis, enzyme inhibition, biochemical uncoupling, lethal synthesis, lipid peroxidations, covalent binding, receptive interactions, immunomediated hypersensitive reactions, genotoxicity, developmental and reproductive toxicity, and pharmacological effects. In 1848, Blank in the United States published his opinion that the biological activity of salt was due to its basic or its acidic component and not to be whole salt. As with lead nitrate, it was the lead and not the acetate or nitrate part. This was for first time a daring thought because it was not until 1884 the Scottish author Brown and Fraser in 1869 made a major discovery. They wrote, "There can be no reasonable doubt that a relation exists between the physiological action of a substance and its chemical composition and constitution, understanding by the latter term the mutual relations of atoms in the substance." This discovery was the first to show structure-action relationships at the turn of present century. Ernest Overton and Hans Meyer independently put forward a lipid theory of cellular depression. This stated that chemically inert substances exert depressant properties on cells, particularly those of a central nervous system, that are rich in lipids and that higher the portions coefficients, the greater the depressant action. The idea that drugs act upon receptors began with John Langley in 1878 in Cambridge. Later, Langley coined the term receptive substances. Paul Ehrlich was already using the term receptor in Germany. In his Nobel Prize address, Ehrlich outlined the receptor as a small chemically defined area which are normally occupied with the cell's nutrition and metabolism but which could take up specific antigens or drug instead. First, the idea of receptor was received with skepticism because of repeated failure to isolate any such substance. However, the idea of receptors became more firmly established by the work of Alfred Clark, who showed that combination of drug with a receptor quantitatively follows the law of mass action. He summed up his work in a monograph a few years before his death in 1941. The period of Second World War 1939 to 1944 was a turning point in the study of structure-action relationship. There was a period when dose-response relationships were highly predominant. Development of physiological and biochemistry also influenced the growth of toxicology. Metabolism of substances was conveniently classified as phase 1 and phase 2 reactions. Concept of QSAR, that is quantitative structure activity relationship. The concept of QSAR was applied to study the toxicity of inorganic cations. While molecules of organic substance reflect their property as a whole, the inorganic compounds dissociate in various degrees and properties have thus to attribute to anion cation or undissociated molecules. Inorganic cations can form complexes with inorganic or organic ligands, contributing new properties to the complex. Component of this system, that is cations, anions, undissociated molecules, could mutually influence each other depending upon the ratio amongst components. Quantitative relationships between the chemical structure of complex and the biological activity form a new line of actions in toxicology. QSAR studies generated the concept of molecular connectivity in order to characterize the organic biologically effective substances. The index of connectivity is deducted from the numeric evaluations of the extent of branching of chemical bonds in the sections of molecule. Exists a correlation between the connectivity index and toxicity of cations. Relationship of toxicology to other sciences. Toxicology is frequently said to be a branch of pharmacology, a science that deals primarily with the therapeutic effect of exogenous substances and with all the chemical and biochemical ramifications involved in those effects. Since the therapeutic dose range of pharmacological compounds is usually quite small and most of these compounds are toxic at higher doses, it may be more appropriate to consider pharmacology a branch of toxicology. Toxicology is clearly related to the two applied biology, medicines and agriculture. In case of medicine, clinical diagnosis and treatment of poisoning as well as management of toxic side effects are areas of significance, while in agriculture, the development of agriculture biocides such as insecticide, herbicide, nematicides and fungicide is of great importance. The detection and management of off-target effects of such compound is also an area of increasing importance that is essential to their continued use. Toxicology may also be considered an area of fundamental biology since the adaptation of organisms to toxic environments have important implications for ecology and evolution. The tools of chemistry and chemical biology since the adaptation of organisms to and progress in toxicology are closely related to the development of new methodology. Those of chemistry provide analytical methods for toxic compounds, particularly for forensic toxicology and residue analysis. And those of biochemistry provide the techniques to investigate the metabolisms and mode of action of toxic compounds. On the other hand, studies of chemistry of toxic compounds have contributed to fundamental organic chemistry, and studies of the enzymes involved in detoxifications and toxic actions have contributed to our basic knowledge of biochemistry. Now, scope of toxicology. Toxicology is the most general sense, maybe one of the oldest practiced science. From his earliest beginning, man must have been aware of numerous toxins such as snake venom and those of poisonous plants. From the earliest written record, it is clear that the ancient had considerable knowledge of poisons. The Greek made use of hemlock as method of execution. More particularly, the Romans made much use of poisons for political and other assassinations. Indeed, it was Dioscorides, a Greek at the court of Nero, who made the earliest known attempt to classify poisons. Although poisoning has enjoyed a considerable very popular at many times and places, the scientific study of toxicology can probably be dated from Paracelsus, who in the 16th century put forward the necessity for experimentations and included much in his range of interests that would today be classified as toxicology. The modern study of toxicology is usually dated from Spaniard or Fila from 1787 to 1853, who at the University of Paris identified toxicology as a separate science. Among his many contributions, he devised chemical method for the detection of poison and stressed the value of chemical analysis to provide legal evidences. He was also the author in 1815 of the first book devoted entirely to the toxic effect of chemicals. Toxicology can be subdivided in variety of ways. Loomis refers to the three basic subdivisions as environmental, economic, and forensic. Environmental toxicology is further divided into such areas as pollution, residue, and industrial hygiene. Economic toxicology is said to be devoted to the development of drugs, food additives, and pesticides. And forensic toxicology is concerned with diagnosis, therapy, and medico-legal considerations. Clearly, these categories are not mutually exclusive. For example, the off-target effect of pesticides are considered to be environmental, while the development of pesticide is economical. Now we'll see environmental toxicology in detail. Environmental toxicology is the most rapidly growing branch of science. Public concern over environmental pollutants and their possible chronic effects, particularly carcinogenicity, has given rise in the United States to new research and regulatory agencies and recently to the Toxic Substances Control Act. Similar developments are also taking place in many other countries. The range of environmental pollutants is enormous, including industrial and domestic effluents, combustion product of fossil fuel, agriculture chemicals, and many other compounds that may be found in food, air, and water. Such compounds as food additives and cosmetics are also being subjected to the same scrutiny. Other subspecialties are frequently mentioned that do not fit into the above divisions. Behavioral toxicology, an area of increasing importance, could be involved in any of these and is usually treated as a separate subspecialty. Analytical toxicology provides the method used in essentially every branch of the subject, while biochemical toxicology provides the fundamental basis for all branches of toxicology. In toxicology, there is some term frequently used. We'll get acquainted with these terms in toxicology as a language of toxicology. Like any other specialized field, environmental health has its own language. Some of the terms may need a few words of introductions, like toxic. A central concept of simply means capable of causing illness. The types of illness caused by environmental toxins are conventionally divided into acute and chronic. Acute illness are those which appear soon after exposure to a toxic compound, last for relatively short time, and then resolve themselves, even if the resolution is in death. The term subacute is also occasionally used to describe disorders with subtle symptoms that are not immediately obvious without special test. Lead workers, for example, often appear to be much healthier than a thorough medical examination reveals them to be a chronic illnesses. By contrast, that may appear years or even decades after exposure, and which may remain unresolved for the victim's lifetime. There are three special kinds of toxic hazards that have special relevance to environmental health: carcinogens, mutagens, and teratogens. As most of us know, a carcinogenic substance is one that causes cancer. A mutagenic substance is one that causes change in the genetic material of a cell. Spontaneous, natural mutations occur in our body cells all the time. The vast majority of them cause no damage, and even when they do, it is usually limited to the lifetime of the cell they occur in. But in rare cases, the cells may continue to grow and divide after a mutagen has altered its basic genetic structure. And if this mutation is passed on to succeeding generations via eggs or sperm, it may cause birth defect, inherited diseases, mental deficiency, increased susceptibility to disease, and a host of other abnormalities and disorders. If the mutated genes are recessive, it may take more than one generation for these effects to show up. Mutagenicity and carcinogenicity are related in same way, but we are not yet sure just how. Radiation is probably the best known example of a mutagenic environmental hazard. Contamination is measured in terms of a concentration of a substance in an environment, and there are a number of different conventions covering the measurement of concentration. The most common system makes use of metric units, particularly the milligram, 1/1000th of a gram, abbreviated as mg, and the microgram, that is 1 millionth of a gram, abbreviated as Ig. Occasionally, in very refined ultra-sensitive measurements, nanograms, 1 billionth of a gram, and picograms, 1 trillionth of a gram, and even smaller units may be used. Obviously, a contaminant concentrations of 1 milligram per kilogram is the same as 1 part of contaminant per millionth part of non-contaminants, or 1 ppm. And this alternative method of indicating relative concentrations is also widely used, particularly for air pollutants, food additives, and pesticides residues. The term ppm and ppb, that is part per billion, are common. ppt, parts per trillion, appears very rarely. Exposure is a way of saying that the contamination in the environment has passed into an organism. A human being is exposed to a toxic compound if some amount of it has entered in his or her body. Exposure does not mean that a person has merely been in proximity of a toxic substance. For example, if you walk past a drum bearing a warning label and containing a toxin, you are not necessarily exposed to whatever it contains. But if the drum leak its content into the air or soil and pollutants the air you breathe or the water you drink, you probably will be exposed to its content. Like illness, exposure may also be subdivided into acute and chronic types. Acute exposure are those that occur over a short period of time, often to high concentration of hazardous substance. Chronic exposures, which are much more common among the general public, and involves longer period of time and for the most part lower concentrations. Dose is the term of measuring that exposure. Basically, the dose a person exposed to a toxic substance receives is dependent on its concentrations in the immediate environment and the duration of the exposure. However, because the interaction of human beings and the environment is a complex, constantly changing process, numerous other factors may also play a part in determining dose. Dose can be a function of weather conditions, the persistence and solubility of the toxic substance in the biosphere, the size of its molecule or particulates, the presence of other compounds in the environment that it may react with, and the age and overall health of exposed individuals, whether the substance is inhaled, swallowed, or absorbed through the skin, and the effectiveness of the body's natural defenses in detoxifying the substance and eliminating it from the body. Some substances like asbestos become virtually permanent contaminant in the body once they have penetrated far enough into the lungs or other organs. Others, such as methanol, are metabolized and excreted from the body in matter of hours at most. The tendency for some substances to collect in a tissue of a living organisms and stay there is known as bioaccumulation. Their tendency to move up the food chain as one species consume another, becoming ever more concentrated as they go, is called as a biomagnification. Traditionally, a threshold was a measurable level of exposure to a toxic substance, below which there would probably be no adverse health effect and above which there probably would be. The setting of safety standard for the work site as well as general environment often involves the assumptions that appropriate thresholds can be determined, monitored, and enforced for toxins in questions. But these assumptions has been subjected to various criticisms in the past few decades. First, it is often pointed out that whether we can measure it or not, it is entirely possible that every molecule of every substance we take into our bodies has some effect on us. It may not be a detectable effect and it may not be harmful or long lasting, but it is an effect. Thus, this argument goes the concept of specific cutoff point below which a substance is treated as though it didn't exist and above which it is considered harmful is misleading. Far more appropriate, proponent of this view argue is the assumptions that these substances have a range of effects beginning at one end and those that are imperceptibly molecular and extending to the catastrophically toxic, ultimately fatal effect at the other end of the spectrum. And LD50 is customarily expressed in terms of a milligram per kilogram of body weight. Thus, a substance whose LD50 is 2 mg per kg is 5 times as toxic as one whose LD50 is 10 mg per kg. In general, substance with LD50 value below 50 mg per kg are considered highly toxic. Those with values between 50 and 500 mg per kg are considered moderately toxic and those with values above 500 mg per kg are regarded as less toxic.